All right, so let's talk a little bit about Unit 5. As I've said with all the units up to this point, the most important thing you can do to prepare for the AP exam is to look over those released multiple choice questions from all the previous secure exams. Your teacher has access to that document. In every Canvas unit, there's a folder for released multiple choice questions, and it will have all of the questions related, in this case, to Unit 5 that have been on the AP exam since 2013. Make sure that you take time to look over that document, figure out why the right answers are the right answers and why the wrong answers are wrong. Again, you can't take that document out of the classroom, but make sure you take the time to look through it. Second most important thing you can do is look over some sample FRQs. And like I did with all the other units up to this point, I've picked 5 or 6 FRQs off of previous exams. I've linked them up right here. These documents contain not only the questions, they also contain the scoring guidelines. So as I've said for all the units, take the time to try to write out an answer to each question. And then once you've done that, compare your answer to the scoring guidelines and use those scoring guidelines to essentially grade your own paper. Figure out what you knew and what you need to work on. The third thing, take time to go through this PowerPoint for Unit 5. Especially pay attention to things you aren't as familiar with. Watch the videos, the screencast, if there's a topic that you're really unfamiliar with. And then finally, if you have some additional time, I've linked up the entire set of Unit 5 student notes right here. So in terms of the most important things to know, this unit's about DNA and the central dogma of genetics. So make sure that you can describe the structure of the DNA molecule. Know what it means that the DNA molecule is anti-parallel. Be able to explain the steps involved in the process of replication and when it happens and why it's important. Also know the functions of the major enzymes involved in the process. Be able to describe what a telomere is and why its function is important. Make sure that you have a thorough understanding of the central dogma of genetics, the process of using DNA as the directions for making proteins. Be able to describe an exception, for example, like a retrovirus, to the central dogma of genetics. Know and be able to describe the 3 stages of the transcription process. Also be able to explain how it happens and why that RNA processing or alternative processing is really, really important. Be able to describe the 3 stages of actual protein synthesis or translation. Be able to use a given DNA sequence to determine the sequence of mRNA that will be transcribed from it, the sequence of tRNA that will be needed to make the proteins, and the actual amino acid sequences that will result. Be able to explain frame shift and point mutations, and be able to explain how that mutations actually affect the structure of a protein and can cause genetic disorders. Be able to explain the purposes of meiosis, and also, maybe more important than that, be able to explain how sexual reproduction and meiosis work together to generate genetic variability, which then acts as sort of almost like a foundation for the process of evolution. Finally, be able to use crossover frequencies to determine the order of linked genes on a chromosome. So this is a gene mapping kind of activity. And there's some sample problems included within this unit. All right, so that's it for our Unit 5 overview.